Okay, so thank you, Kathy and Ron, for having me, and thanks to everyone here for waking up so early in the morning. And it's hard to get to a talk at 8 a.m. And so, as Kathy said, our lab is really interested in this interaction between drugs and the microbiome. And I'm going to tell you a little bit how we're trying to, take this really complex series of interactions and start drilling down to mechanism. But from a really high level, the questions that we ask in our lab, I think, are really similar to the question that many of us in the room are fascinated by. That's a sort of age-old question in pharmacology, which is, why do drugs work, miracles for one patient, whereas for the next they might have little to no impact? And is that due to, variation in environmental risk factors, or due to genetics, changes in the human genome? But one of the things that's been really striking by thinking about the microbiome, or the trillions of microbes that live in and on the human, is that this really expands our concept of the human genome by multiple orders of magnitude. And so just to sort of give you a sense for this, if you take a given cell, a human cell that's found in the gut, like an enterocyte, there's on the order of 25,000 genes. Whereas if you take the microbiome just within the gut, there's at least millions, maybe tens of millions or even higher, unique genes. And I think this is both a daunting number and very inspiring in that we know a tremendous amount about every gene in the human genome, but we know almost nothing about the average gene in the microbiome. And so, you might wonder whether or not all this genetic diversity in the microbiome actually matters. Maybe it's just sort of some baggage we're carrying along that doesn't actually do anything interesting, and so we don't have to worry about it. But hopefully, I'll convince you over the next 20 or 30 minutes that they actually do matter. And one of the reasons why genes found within the microbiome are important is that they encode a lot of enzymes, and they encode far more enzymes than are found in the human genome. And these enzymatic machinery that are found in bacterial cells, as well as other microbes, are capable of making a lot of compounds that are unique to the microbiome, so they contribute maybe the majority of small molecules that are found in circulation. And they also are sort of master chemists at changing the chemicals that are in our body. And so they can take drugs and turn them into new types of chemicals. They also change components of our diet. And they manipulate a lot of the endogenous compounds that are in our body. And so our metabolome is really tightly linked to the microbiome. And so, we sort of think about this interaction between the microbiome and pharmacology along, the classical paradigm of ADME. As Kathy mentioned, we're interested in our lab in how the microbiome controls drug transporters and both the absorption and efflux of drugs back into the intestine. We're interested in distribution, so how the microbiome might remotely control the entry of drugs into other tissues. How do they change the amount of drug that makes it into the brain or into the lung or another organ? And many of the other labs in the field are really focused on excretion. So, this ability for the microbiome to control the clearance of drugs. But probably the most well-studied area is metabolism, and so that's why I put the capital M. And that's what we're going to talk about today. So this is, by far what we know the most about right now in terms of the role that the microbiome plays in pharmacology. Okay. And in fact, we know so much about microbiome and metabolism of drugs that there's been books written. And so I don't know, some of you in the room might have read this book. It's really fascinating. I didn't write it. I promise I'm not affiliated. But it's a story that I'm going to briefly summarize in the next slide that's really remarkable. It covers the first antibiotics that were ever used in patients. It includes things like Nazis and microbes, and it's just a really fascinating story. So I recommend it. Okay. And so just to sort of briefly, spoil the book for you, there's a fellow named Gerhard Dummack, who was a researcher at Bayer back in the early 20th century. And Gerhard, like many people, was trying to kill bacteria. Unlike today, where we think about this relationship with bacteria as being much more complicated. At the time, researchers were obsessed with trying to kill off microbes as fast as possible. He was actually awarded the Nobel Prize for this really seminal work that showed that azo dyes, which are these compounds that have two nitrogens double bonded to each other, have antibacterial effects. And this led to Prontosil, which is the first sulfonamide drug. Okay. But the thing that's really fascinating to us as microbiome researchers is that it turned out that Prontosil itself is actually a prodrug. And so what happens is, if you administer Prontosil, to a patient, it gets cleaved into these two different downstream metabolites, and the active compound is actually sulfanilamide. And it turns out that there's no enzyme in the human genome that can perform this reaction. It requires bacteria. And so, from the very beginning of antibiotic discovery, there was this realization that we need the microbiome to actually activate some of the prodrugs that are given to patients. And so we wondered, whether or not the opposite is also true. Is it possible for active compounds that are given orally to be intercepted by the microbiome and inactivated, limiting the bioavailability of the active compound? And in fact, that was proven about 50 years after Gerhard's discovery. And this really focused on work around the cardiac drug Digoxin, which is sort of the poster child, as many of you probably know, for a drug that's inactivated by the microbiome. And so I'm just going to sort of walk you through the background for Digoxin. And so Digoxin is really fascinating because it's one of the first natural products used in medicine. It's isolated from the foxglove plant shown here in my father-in-law's garden. And it's given as an oral drug. It's incredibly toxic because the target of Digoxin is the sodium-potassium ATPase, which is conserved in all cells, both microbial and human. But it's useful in patients that have heart failure and cardiac arrhythmias, and is still widely used around the world. But one of the things that was discovered in the 1980s was that about 10% of patients have really high levels of this inactive metabolite called dihydrodigoxin. And one thing to note is that the dihydrodigoxin, the metabolite, is incredibly similar to the drug. There's only this double bond shown in the lactone ring that's reduced. And so that tiny chemical modification is enough to change the activity of the drug, meaning that it no longer binds to the target in the heart or other cells. And so it's both inactivating the drug as well as detoxifying or helping to clear the active drug from the body. And so John Lindenbaum, as well as others like Les Bennett at UCSF, did a series of studies in humans. And what they found is that if you look at a patient that's taking broad-spectrum antibiotics, there's increased bioavailability of Digoxin. And so this, suggested that maybe there's some bacteria that's actually metabolizing the drug. But the thing that really got us excited was that the Lindenbaum lab screened hundreds of different gut bacteria and actually found that, unlike some of the other drugs we're studying now, there's only a single bacteria that can perform this reaction. And that bacteria is named Agarthala lenta, which I'm going to tell you a lot about in a couple slides. And so this is a really nice example where, of the hundreds or thousands of different bacteria that live in the gut, there's just one type that performs the reaction. And so, this was all super exciting, led to a lot of speculation about the sort of future of microbiome in pharmacology and personalized medicine. But the problem was when, people tried to translate this discovery, it turned out not to be so useful. And so this was a nice sort of quote from Ian Rowland in a book he wrote in 1988, where he says, E. lenta is the organism known to perform the reaction, but neither the presence of these bacteria nor their concentration in the gut relates to Digoxin reduction. And so it turned out, by the best methods available in 1988, you couldn't predict anything about drug levels based on knowing whether or not E. lenta was there. And so, why is that? Is it because, we should just work on the human genome, or do we just not understand enough about the microbiome? And so Henry Hazer was a postdoc that joined our lab back in about 7 or 8 years ago. And I'm just going to summarize his work while he was in the lab. And Henry was able to identify sort of two factors, which we think are two of many, that contribute to the variation between people in whether or not their microbiome will metabolize digoxin. And so those two factors are very subtle strain-level variations in the Elenta population, so genetic differences between Elenta strains, and interactions between these bacteria in the diet. So just to sort of walk you through this in a really simple cartoon, again, we think about digoxin getting inactivated, forming this downstream metabolite dihydrodigoxin. Henry was able to find two genes within the Elenta genome that were highly upregulated in response to digoxin, which we named the cardiac glycoside reductase operon, or CGR1 and 2. And he found that the type strain of Elenta, which carries these genes, is capable of performing this reaction. We looked at two other strains of Elenta and found that they were missing these genes and that they were not able to metabolize the drugs. And on top of that, we showed in mice that were raised in a way that they were sterile prior to being colonized with only Elenta, that if we changed the amount of protein in the diet, we could control this activity. And that was, we think, due to changing the levels of arginine in the gut, which acts as a potent inhibitor of this activity in vitro. Okay. But there's a few sort of, I guess, important questions that remain to be answered about this system. And this is going to be the work that has been led by Nitzan Koppel, who's a graduate student in our collaborators lab, Emily Balskos at Harvard, and Jordan Bassan's a postdoc in our lab. And so the first basic question was, are these genes really necessary and sufficient for drug inactivation? Do you need other genes in the Elenta genome, or is this really the only genetic machinery that you need? Another important question was, are they prevalent in the human microbiome? And so, the type strain of Elenta, interestingly, was isolated back in the 1930s. And so we didn't know, because that was the only strain that we had available at the time, we didn't know whether or not current humans are even colonized by bacteria that have these genes. And another sort of really important question is whether or not these enzymes are specific to the drug. So does the microbiome, has it evolved to specifically metabolize drugs, or is it intending to target something else that's normally in the body and accidentally metabolizing the drug? Okay. And so, before I sort of get into the answers to these questions, I want to just tell you a little bit more about Elenta. And so this is a bacteria that probably nobody in the room has heard about. I hope in a few years, all of you will have heard about it. But it's a really neglected member of the gut microbiome. It's probably the closest relative that you guys have heard about is Bifida Bacteria, which is widespread in yogurts or probiotics. And it's shown here in a couple images. So you can see on the left here, this is a high-resolution view of Elenta grown in the lab. You can see it forms sort of these clumpy, all of these cells are Elenta, but they sort of grow closely associated with each other. And then the video here on the right is actually Elenta growing in real time, and you can see that they sort of like form these long chains. It doesn't form spores, so unlike Clostridia that you might have heard about, it's an obligate anaerobe, so it's incredibly sensitive to oxygen, and it's a member of this Actinobacterium phylum, which includes the bifidas in your yogurt. We have found that it's incredibly prevalent in humans, so maybe as high as 80% of the people around the world are colonized with Elenta, but at very low abundance. And it's really famous, in our circles for being a master of metabolism. And so it can affect drugs, other foreign compounds like dietary small molecules, and a lot of endogenous compounds like bile acids and hormones and neurotransmitters. It's also been recently linked to disease, and so this is an active area of research in our lab. We're interested in, the potential role Elenta plays in sepsis or bacteremia, as well as its more controversial links to diabetes, rheumatoid arthritis, and other autoimmune diseases like MS. Okay. But the major challenge in studying Elenta is that we don't have any genetic tools for these organisms. And secondly, that there's really just a handful of Elenta that have ever been isolated and studied in the lab. And so this sort of challenge led to one of the interns in our lab to draw this sort of Pokemon cartoon, where, I don't know if anyone's a Pokemon Go fan, but, we think about running around the world and trying to catch bacteria instead of Pokemon. And so this is, gotta catch them all. We're still working on this, so if anybody in the lab has, a freezer full of Elenta, let me know. But we've so far collected 30 strains that come from 6 countries, 3 continents in North America, Europe, and Asia. They interestingly come from different samples, and so like I said, Elenta is often isolated from bacteremic patients. So we have blood Elenta, stool, and cancer-associated Elenta. And they also span 7 decades, and so we can look at the Elenta genome as far back as 1935. We've also included some close sort of sisters of Elenta, which is Paraagarthella, Gordonibacter, and Asaccharobacter, that are closely related evolutionarily. And a lot of this collection really benefited from Emma Ellen Vercoe, a scientist up in Canada who's sort of an expert in isolating new and interesting bacteria. Okay. And so one of the things now that we can do with the strain collection is just to genome sequence all the strains and compare them to each other. And so this is just a view of the global nucleotide identity across the strain collection. We've included both the 30 isolates that we're working with in the lab as well as other genomes that we can pull out of the databases. And what you can hopefully see is that there's this big red block which represents an almost 100% average nucleotide identity, which is the Elenta species. And then, these sort of relatives of Elenta are all quite different from each other. There's a few little clusters. And so we can now, in an unbiased way, define the Elenta species just based on genomic similarity. And it turned out that the sort of classical definition was actually pretty good. And so this is, if we compare within things that are called Elenta, you can see they're almost completely identical at the nucleotide level. And then if we look at, comparing those Elentas to other species, they're much more dissimilar, around like 85% identity on average. And so this is now sort of our genomically defined Elenta species. One thing that's sort of interesting is that you can now, build genome phylogenies across these different strains. And so these are all within this group of Elenta. But you see they sort of form multiple clusters. And so there's maybe one sort of subspecies here, and then a second group on the top. Okay. And so all of this was sort of to now allow us to go back and look at the Elenta genome and see, what parts of the Elenta genome map to function. And you can do this for really any activity you might be interested in. I'm going to tell you about today how we've used it to look at digoxin. And so we screened all the isolates using mass spec for the ability of those strains to metabolize digoxin to dihydrodigoxin. You can see in red, these are digoxin-reducing strains. So we now have 8. And then the blue bars represent strains that don't significantly metabolize drug over background. We can now compare the genomes of those 8 reducing strains to the non-reducers. And so we've sort of ranked all the genes found within the Elenta genome based on how well they match this pattern of metabolism. And what we find is that there's a single gene cluster, shown at the top here, that's in all of the metabolizers and missing in all the non-metabolizers. And so it perfectly matches the function. And so what is this cluster? It turns out that it contains the 2 genes that I told you about earlier, CGR1 and 2. But also contains some neighboring genes. And so all of these strains at the top here have this entire region of the genome conserved, whereas these genes are missing in the strains found at the bottom, but the flanking regions are conserved. And so what's in this gene, again, CGR1 and 2 are there, and I told you before that they're really highly induced in response to digoxin. Whereas the rest of the genes in the region are really expressed at low levels normally. One of the genes is maybe slightly upregulated, but there's not much transcriptional activity. And so we thought, this sort of now narrows down to see either CGR1 or 2 as being the important enzyme that's necessary for this reaction. And so that's where Nitsa and Koppel did a heroic job of troubleshooting different ways of cloning these genes from Elenta into model bacteria where we have genetic tools. What she found is that, if she put in an empty vector with no gene insert or CGR1, there was no metabolism of digoxin. But if she clones in CGR2, now that single gene is capable of enabling these bacteria to perform the reaction. And we see sort of similar levels in both genes. And so this is super exciting because it shows us that CGR2, this one gene we've identified from Elenta, is capable of this reaction. And so what do we know about CGR2? It has a number of sort of interesting domains. The first is a secretion tag. So this predicts that this is actually a secreted protein that Elenta is releasing outside the cell. As well as a number of motifs that suggest that it binds flavin. And so this suggested something very simple that was testable, which is that we should be able to add flavin to the purified CGR2 protein and enable it to metabolize digoxin. And so we tried that, and much to Nitsan's chagrin, when she adds either type of flavin to purified CGR2 protein, there is absolutely no activity. And so the protein was broken for reasons we did not understand and couldn't really be predicted computationally. And so what she thought is that, a lot of reductases that are encoded by anaerobes contain these really oxygen-sensitive iron-sulfur clusters. There's ways of reconstituting these clusters just by sort of bathing the protein in iron and sulfur. And so she found that after reconstitution in the presence of FAD, now the protein activity turns on. You can also kill the cluster, and so if you boil protein or add oxygen, you actually destroy that part of the protein, and that also got rid of the activity. Another sort of hint that this cluster was there was just based on absorbance, and so there's sort of a characteristic signature found in the reconstituted protein that's also diminished after adding oxygen and isn't found in that original prep that hasn't been reconstituted. And then finally, she used electron paramagnetic resonance spectroscopy, which is sort of the definitive approach to prove that there is multiple iron-sulfur clusters in CGR2. Okay. And so the other thing we wondered, going back to the genetics of the system, is whether or not this gene is the same across all Elenta, or are there genetic polymorphisms within CGR2 that matter for function? And so shown here is the whole cluster across the 8 different isolate genomes. You can see the triangles, the red triangles represent substitution. So these are single amino acid changes in the protein. You can see, one of the striking things is there's basically no changes here. There's just a few across the entire region. Within CGR2, there's only 2 amino acids that we ever find changed. And then, we asked whether or not these changes were also found in metagenomic data, so shotgun sequencing data from human gut samples. And in analyzing those metagenomic data sets, we found that we confirmed these 2 substitutions, and we found a few additional ones, but you'll note that this region is really highly conserved. And so the one that we were really interested in was this Y33N, which is variable both within genomes and within microbiomes. And so we went on to look at strains that vary at that position. We found that if you actually just took whole cells, so this is the original strains, with the Y version versus N, that there was a slight but not significant difference in metabolism. And obviously there's many other differences between these strains that could confound that comparison. And so to do a more controlled head-to-head comparison of the 2 versions of the protein, we purified both and then measured the rate of the reaction. And you can see that Y is a good enzyme, whereas the N version is very slow at metabolizing digoxin. Interestingly, we find if you sort of wait long enough, even the N version will eventually metabolize the drug, but at a much slower rate. And so it's not a full loss of function, it's a partial defect. Okay. We also find that despite this conservation, that CGR2 is really different from other reductases. And so this is sort of a global view of all the reductases that are found in gut bacterial genomes, annotated by their major substrate. And so the fumarate reductases are shown in blue, ketosteroid dehydrogenases in yellow. And our favorite reductase, CGR, is the single dot all alone. And so it's completely different from any characterized reductase based on its both nucleotide and amino acid sequence. And so next we wondered, is this new reductase, even though it's different from these other reductases in terms of its sequence, could it still accept things like fumarate or ketosteroids as a substrate? And so is it, trying to metabolize something that's normally in the body and sort of acting in a promiscuous fashion to target drugs? And so, now that we had purified CGR2 protein, we could screen in vitro a large panel of substrates. And so shown here is the rate of the reaction when we add different cardenolides. So this is the broad family of compounds that contains digoxin. You see that all of them are substrates, some slightly better than others. But what was really striking is that all of the other related compounds that are normally in the body were not substrates. And so we don't see any activity against the normal endogenous compounds that we think could have been putative substrates for CGR2. And so this activity of CGR2, at least in this in vitro system, is really restricted to these toxic plant compounds like digoxin. And so you might think that, based on this sort of weird and quirky activity, that these genes would be really rare, based on the fact that there's no obvious function for them in the human microbiome. But surprisingly, they're actually quite prevalent. This is just prevalence as a function of abundance from metagenomic data. You can see by this assay, Elenta is in about 40% of people, whereas this gene cluster is in about 25% of people that have been sequenced. And so it's quite widespread both around in America as well as in other countries. We can look at the correlation between CGR2 and Elenta, and not surprisingly, there's a strong correlation consistent with the fact that these genes are really unique to the Elenta species. But what's interesting is that when samples fall off the diagonal, you see that they're found below, like down here, all these dots. And so that suggests that these are samples where only a fraction of the Elenta cells carry the gene. And that's consistent with other data we have previously published, that people, each of us sort of harbors a mixture of different Elenta strains. And so there's both CGR positive and CGR negative strains that are competing within the gut. And so at the sort of biochemical level, this is how we think about the system. I didn't really talk very much about CGR1, but it's a membrane protein that we think shuttles electrons to CGR2 through some sort of protein-protein interaction, which then uses these iron-sulfur clusters to catalyze the reduction of digoxin as well as other plant compounds. And I think the most surprising thing that came out of this work was this idea that the microbiome might protect us from these toxic plant secondary metabolites. And so I think a lot of us, including myself, prior to this had hypothesized that the main role of this system would be to work on endogenous compounds. But surprisingly, this enzyme seems very specific for cardenolides. And so there's a lot of sort of interesting things we could think about doing in the future. One is, can the microbiome actually metabolize enough digoxin to rescue from overdose? So say if you were poisoned with a foxglove, would it be able to save you? We also really wonder whether or not there's a direct benefit of this for Elenta, or is our microbiome just protecting us from toxins in an altruistic fashion? Or maybe there's an endogenous compound we just don't know about, and open to any ideas that you guys might have. And then finally, this has really raised the question of whether or not a similar mechanism evolves in microbial communities that are constantly exposed to cardenolides. And so together with Denise Deering and Mark Hunter, we're in the early stages of a sort of survey of environments where cardenolides are normally found. And so this is both in the foxglove itself, in monarch butterflies or caterpillars that consume these plants. Lophiomes, the strange rat species that eats cardenolides, and even in human patients. We're also excited by the possible translational implications of this type of work, and so we're working with clinicians at UCSF and elsewhere to try to develop diagnostic tests where we could pair information about the microbiome with personalized dosing regimens. And we're also very interested in developing strategies to inhibit these systems. And so could we use arginine, like I told you about for digoxin, a dietary supplement to block this metabolic activity? Or maybe we could develop other small molecule inhibitors that inhibit this in a way that doesn't affect other systems in the body. In addition to that, we're actively trying to expand into other areas, and so digoxin is just one of many drugs that's inactivated in a similar fashion by the microbiome. For example, 5-fluorouracil is also reduced by bacteria, forming an inactive metabolite. And so we're really interested in, just as we've done for digoxin, trying to understand exactly how the microbiome is impacting each of these medications across a wide range of different diseases. And so to close, I just want to leave you with a few sort of very basic takeaway points. So the first is that the microbiome contributes to these inter-individual differences in ADME. It may not be the most important factor, but we think it's really important if we were going to have a comprehensive understanding of pharmacology to understand both the host and the microbiome. I think one thing that we hear a lot is that the microbiome is so complicated, how can you ever understand mechanism? And so hopefully I've sort of showed you today, hopefully not in too much detail, that it's possible to take these complex systems and distill them down to mechanism. But just like with the human body, the microbiome is incredibly complicated. And so we need to not only understand it, but also its interactions with the host and with environmental factors like diet. And finally, this is just the beginning of a really new area of study. The microbiome field as a whole has really only been around for about a decade, and this area of sort of pharmacomicrobiomics, or the intersection between microbiome and pharmacology, is really just now beginning. Okay. And so if any of you, are interested in this area, I would really encourage you to check out this Keystone Symposium. I'm organizing it together with Emily, our collaborator, as well as Dennis Wolin down at Scripps. And it's really going to focus on the sort of broader intersection between biology and chemistry as it relates to the microbiome. And so hope to see you there. This is just me as the biologist staring at these drugs in wonder while Emily judges me. But with that, thanks again for having me, Kathy, and I look forward to any questions.
Thank you. Thank you, Peter. Okay, that was a great talk. Are there questions for Peter?
Any age effects? Just in general on the microbiome, or? Yes, yeah, so there's a lot of work in this area has been done by my thesis mentor, Jeffrey Gordon, and he's been very interested in sort of the development of the microbiome in early life. And what they found is that there's a period of about 2 to 3 years when our microbiome becomes established, where there's a lot of changes, but largely after 3 years and into adulthood, there's some degree of stability, although there's still a lot of dynamics. And so there is a very important window where the microbiome becomes established. Later in life, it becomes more complicated. There's some evidence that maybe in the elderly there's a change in the microbiome, but that could potentially also be driven by environmental pressures. So, changes in lifestyle, changes in diet, changes in, where you live.
Thank you so much for sharing this research. It's really impressive. So my question is, I wrote it down, it's kind of long. The current molecular testing is very much focuses on the patient's genetic profile. And here you have shown that there is an absolute gap to understand the drug response. So first of all, when will the gut microbiome test will be ready, and how to merge these two tests to each other. Is there such a research going on?
Yeah, those are all great points. And I think, one of the challenges, I guess, that I talked about at the beginning was, for the microbiome, there's so many more genes than for the human genome. And so that's both exciting in terms of basic science. We're going to discover a lot, interesting new enzymes. But we also often deal with the problem that there's a lot of redundancy in the system. And so even if I had a great genetic test for gene A, there's a lot of other genes that are important that I can't measure. And so it's a big challenge. And I think, what a lot of us are trying to do is find examples as sort of initial case studies where there's a smaller scope of genes that are important for a phenotype. And so maybe, for digoxin, that's a more tractable example where, even though it's complicated, there's a very conserved gene that's involved. And we're trying to find other examples like that. In terms of actually translating it, we're looking for partners. I think, we're collaborating with some groups, but if anybody here is interested, I'd love to look at that. And what we don't really have in the field so far are paired studies where we have, the best diagnostic tests in terms of, blood or human genetic parameters, as well as, the sort of gold standard microbiome tests. Yeah.
Okay, 2 more questions here and then here.
In looking at the tyrosine and asparagine variant that you had in the enzyme, did you find some individuals tended to have a higher frequency of one versus the other, or certain populations had one versus the other? Was there some sort of change or difference between people?
Yeah, that's a great question. So, yeah, we do find, we don't have the numbers, I think, to look at population level differences, but we definitely find that, some people are dominated by one variant, whereas others are dominated by the other variant.
So do you think there may be a reason for that, or just chance?
Yeah, it's a great question. And, it's really maddening because, that variant is really bad at metabolizing the one thing we know the enzyme can act on. So, it potentially suggests that there's other substrates that we haven't measured yet where that variant is actually useful for. Or alternatively, maybe it's just a loss of function mutation that's eventually going to be weeded out of the population.
Okay, final question.
Yeah.
So you sort of brought this up, but the variance in the other direction. So is there some other mechanism by which this drug is being chewed up in the gut, or are there other bacteria that we don't know about that have a operon with a homologous function, or a equivalog function?
Yeah. We've looked pretty. We've been trying to develop more sophisticated ways to do this, but so far all of the evidence shows that, for this particular gene, it's unique to that species. That definitely doesn't mean that there could be similar, enzymes that have the same activity that are just unrelated and have no genetic similarity to this gene. And so, we are interested in looking at, in the network I showed you guys, a lot of those sort of orphan reductases are in the microbiome, but we don't know the substrate.
And just to make sure I remember the manuscript correctly, there were people with very low digoxin levels who did not have the CGR operon. Is that correct?
So there were people that had. Well, I don't know which figure you're talking about, but if you're talking about the sort of, we do these incubations where we'll take stool samples and add drug and then measure reduction. And there's definitely not a perfect prediction based on gene level.
Thanks.
So there might be other regulation. Yeah.
All right, let's thank Peter again for a great talk. Thank you.